Okay, so for today, I'll be discussing to you about organogenesis, which is lecture number 7 for our alignment of topics. So to start off, organogenesis is the mechanisms of in invertebrates are actually similar, but the body plan is very different. So when you say the body plan, of course, this is the axis formation of different animal models. So as you all know, there are some cells that do go dorsally or ventrally. Some other cells would actually go posterior or anteriorly. So the basis of this organogenesis is very different when it comes to the body plans of different species. And for example, the neural tube develops along the ventral side of the embryo in invertebrates rather than dorsally as it occurs in your vertebrates. Okay? So for morphogenesis in animals but not plants involve movements of cells. So majority of the cell signals that is being utilized are more likely to happen when it comes to animals and not in plants. Okay. So for the mechanism of morphogenesis, first there is reorganization of the cytoskeleton, and it is a major force in changing the cell shape during development. For example, in neurulation, microtubules oriented from the dorsal to ventral in a sheet of ectodermal cells help lengthen the cells along the axis. So this is an example of an organogenesis that's happening in a chick, which is actually quite similar to that in frog. So as you see here, you have the different movements of the ectodermal cell layers. So again, the blue ones are represented by the ectoderm. The red cells are represented by the mesoderm, and then the yellow ones are for the endoderm. Okay, and then you have your coelom here, and then the developing somites, and then in the middle you will see the neural tube, and then the notochord, which will eventually become your spinal cord, and then the primitive gut, which is your archenteron, and then of course the lateral foldings, okay, which actually pinches and separates the dorsal side of the neural plate or the neural formation. And then this is the part of the yolk, and then here the yolk sac, the yolk stalk, and the um, combination of the three extra embryonic membranes. So this is how organogenesis actually happens initially to chick embryos. Okay, so this is another representation of the late organogenesis that's happening in the chick embryo. So you have here, as you remember or recall, the different cellular changes and development when it comes to the different animal models are very very different from each other. Okay, so in here for the chick, you see that eventually it will already grow its eyes, the otic capsules, and then the forebrain, the heart, bulging heart, and then the blood vessels would be more vascularized for the chick. And then there's also the formation of the somites and of course the neural tube. So comparing from what you have seen a while ago from the early organogenesis, which is apparently happening after the neurulation part, eventually with the late organogenesis, you will already see the development of the distinct parts of an animal model. Okay, so in here we have the chick embryo as an example. So change in cell shape during morphogenesis. So again, from just a flat ectodermal layer from the neurulation process, of course, you will develop the neural plate. So as you see here also with the microtubules or the different cytoskeletal components of this cell, they also do different, or they also conform a direction on how they will be arranged. Okay, so from the neural plate, you will then eventually form the neural groove. Okay, so in here you will also see that the actin filaments are also having its certain direction to be able to, support the different cells, and then eventually this part will already be closing, and then the neural tube will already be produced or developed, and then the neural crest cells would eventually um, separate, okay, from the external part of the neural tube or the neural plate, and then eventually forming the neural tube. Okay, so other than the position of the cells, there's also a change in the shape of the cell itself. Okay, so convergent extension of a sheet of cells. So the cytoskeleton promotes elongation of the archenteron in the sea urchin embryo. And then this is convergent extension, which is the rearrangement of cells of a tissue that cause it to become narrower and longer. Okay, so from a short type of cell, what happens is that there's a convergent extension, and eventually the arrangements of cell, makes it narrower and longer. So instead of the cells, stuck together, they eventually become, the same length and size at the end of the process. So convergent extensions occur in the developmental process, and the cytoskeleton also directs the cell migration. Okay, so when you say convergent, di ba, in Tagalog, parang nagbubungguan sila. And then eventually, once they already have the opportunity to, embed with each other, and you move the plates or the cells within each other, tignan nyo yung nangyayari sa kanila. No? Eventually, nagiging parang alternate yung arrangement ng cells. Okay? But this is just to give you um, a look on how the convergent extension process happens. Okay? So next is the programmed cell death or apoptosis. It at various times during development, individual cells or set of cells or whole tissues stop developing and are engulfed by neighboring cells. So for example, many more neurons are produced in developing embryos than will be needed, and extra neurons are removed by apoptosis. So it is very important that there is what you call this apoptosis. Again, as what I have mentioned before, if there's no apoptosis that's happening, especially in the process of organogenesis, you will eventually, of course, abnormalities would actually arise. It just means that hindi tumitigil kasi yung pagpo-produce or pag-move ng mga cells na yon. So if those migration or redevelopment or reproduction of cells won't be stopped, of course, the possibility of having extra limbs or extra part of the body would be possible without apoptosis. So apoptosis is not, a negative process itself, but then again, it regulates, no, the amount of cell that develops inside the body to be able to have the ideal um, cells or position of cells in a developing model. Okay? So, cytoplasmic determinants and inductive signals contribute to the fate specifications. So, when you say cytoplasmic determinants, of course, this is the components of your cell wherein the organelles that lay on the cytoplasm are involved. Okay? So, and then next, when you say inductive signals, of course, these signals are being used for cell-to-cell communication. Okay? So the determination is the term used to describe the process by which a cell or a group of cells become committed to a particular fate. So for example, if that cell is, the cells for the head, of course, that would be the determining type of cells that should be present in the side of the head. And then for differentiation, it refers to the resulting specialization in a structure and function. So as we have discussed before, in the beginning, we only have generalized types of cell, right? So once this organogenesis already happens, there's always a determination to where these cells would go to, and then once those cell um, reaches the position or the direction where they should be, then differentiation happens. So for example, in the head, so once a group of cell goes to the head, ano namang cell ngayon yung pwedeng ma-develop, or ano namang organ yung pwedeng ma-develop ng mga cells na yon. It can be cells of the brain, cells of the neurons, it can be also cells of the ears, depending on what would be the fate, okay, or what would be the specialized structure that they are intended for. Okay? So cells in a multicellular organism share the same genome. So when you say same genome, it's like a DNA backbone. Okay? And then differences in cell types are the result of the expression of the different set of genes. Okay? So for example, humans, of course, all of us same the shared genome. It's just that we actually have the differences when it comes to the expressions of these genes. So, hindi naman tayo lahat magkakamukha. We have differences because of genetic variation. Okay? So, some would have um, curly type of hair. Some would have different skin complexion. So, that's how, the gene expression happens. Okay? So, as also in plants, okay, so in plants we have what you call, 'di ba, pag nag-grouping tayo ng mga plants, to be able to say that a certain type of plant belongs to a certain type of family, there should be a distinct characteristic that they share within a pool of plants. Okay? So fate mapping. So fate maps are diagrams showing organs and other structures that arise from each other, from each region of an embryo. So classic studies using frogs indicated that cell lineage in germ layers is traceable to blastula cells. So we already know that fate maps can be, um, can be done using different types of experiments, right? So, yun yung pwede tayong gumamit ng mga dyes, and then we observe them under the microscope for a given period of time, and then makikita doon kung paano nagmo-move yung mga cells na yon. Okay? So, fate map of a frog embryo. So, from a blastula, again, the three different colors that we usually use is blue for the ectoderm, you have the mesoderm for red, and then endoderm for the yellow. So, coming from a blastula like this, eventually it will change into this neural tube stage, which shows you another transverse section. So from this epidermis, it will eventually look like this, and then, the central nervous system would detach from the epidermis itself. And then the mesoderm, coming from the lateral part, it will eventually go into a circular motion also, and then eventually would give you, or would give rise to the notochord. And then the mesoderm would be, or the endoderm will also be lodging within the neural tube stage. So, pumapasok siya sa loob. Okay? So, cell lineage analysis in a tunicate. So, for in a tunicate, so this is another example. So, if this is a 64 cell type of embryo, and then blastomeres are actually injected with dye, so a portion of the set of the embryo has been injected with dye, and under the microscope, it would actually look like this. So, this is actually an example of how you trace fate maps of the cell movement. Okay? So, for example, if this 64 cell embryo has been injected with a dye on this part, what happens to the cells are actually this one. They develop into this dark pigmented part of the tunicate. So, if the dye is also injected to another part, So this one gives you the idea to where the dye or the cells that have been dyed uh, goes to. Okay. So later studies of Caenorhabditis elegans use the ablation or destruction of single cells to determine the structures that normally arise from each cell. So the researchers were able to determine the lineage of each of the 959 somatic cells in a worm. Okay? So the cell lineage in Caenorhabditis elegans. So this is actually a representation of one zygote. So for the first cell division, what organs were actually developed. So this one is a timeline. So from 0 hours to 10 hours. So basing the development on the timetable itself, mabilis na nagde-develop yung nervous system nila, the outer musculatures, the gonads, outer skin, the germ layers of the gametes, and then before hatching, tsaka palang mag-develop yung intestine nila. Ayan. So, of course, the movement of cells, again, it can be anterior to the posterior. Okay, so this is just a representation of the cell lineage in a Caenorhabditis elegans. Okay. So for the germ cells are the specialized cells that give rise to the sperm or eggs. Ito yung mga tinatawag natin ng mga general types of cells na meron tayo. So complexes of RNA and protein are involved in the specification of each germ cell fate. So for example, in C. elegans, such complexes are called P granules that persist throughout the development and can be detected in germ cells of an adult worm. So, determination of germ cell fate in C. elegans. So, as you see here, we have P granules, which are distributed throughout the newly fertilized egg and move to the posterior end before the first cleavage division. So, with each subsequent cleavage, the P granules are partitioned into posterior most cells, and P granules act as cytoplasmic determinants, fixing germ cell fate at the earliest stage of the development. So, usually yan mga P granules. Okay? So, partitioning of the P granules during C. elegans development. So, for the granules, which were, injected with fluorescent dyes for them to be able to observe the movement. So, for a newly fertilized egg, as you see, scattered yung mga P granules niya. But once the zygote already reaches the prior to first division, kung makikita nyo, nasa isang side na lang yung concentration of the P granules. So once the embryo becomes 2 and lumilipat sa isang side, and then the 4-cell embryo, makikita nyo na isang side na lang talaga yung mga P granules nila. Okay? So, yun nga, as what have been mentioned, the direction of these P granules are going posteriorly. Okay? So, axis formation. So, a body plan with bilateral symmetry is found across a range of animals. So, when you say bilateral symmetry, what you see on the right side can also be seen on the other side or the left side. So, this body plan exhibits asymmetry across the dorsal ventral and anterior posterior axis. So, if you know your basic anatomical or positions, you should know what is dorsal and what is ventral or anterior posterior axis. So, the right and left axis is largely symmetrical. Okay? So, for the axis formation, the anterior posterior axis of the frog embryo is determined during oogenesis. And then the animal vegetal's asymmetry indicates where the anterior or posterior axis forms. You know that the animal and vegetal asymmetry would actually depend also in the amount of yolk, right? So depending on where the animal pole or the vegetal pole is, it will tell you what is or where is the anterior or the posterior part is. Okay? And then the dorsal ventral axis is not determined until fertilization. And upon fusion of the egg and the sperm, the egg surface rotates with respect to the inner cytoplasm. So we have already discussed how the different, rotations happen when it comes to the development of the zygote. Okay, so this cortical rotation brings molecules from one area of the inner cytoplasm of the animal hemisphere to interact with molecules in the vegetal cortex. And then this leads to the expression of dorsal and ventral specific gene expression. So it's very important that we know the axis formation of a certain animal model for us to also make sure that the direction of the different cells within that body plan is actually going to the right position. Okay? So the body axis and their establishment in an amphibian. So again, when you say anterior, usually it is on the side of the mouth, and then the posterior is the anal part. And then you also have the right and then the left side, and of course the dorsal and then the ventral. So pag sinabing dorsal, guys, ito yung gawin nating analogy ang human. So pag dorsal sa likod, pag ventral sa dibdib. Okay? So the three axes of a fully developed embryo, again, for an establishing axis, we always have what you call the animal pole, which is usually the darker side of the embryo, and then the vegetal hemisphere or the vegetal pole. So once there's already a fusion or a point of fertilization or entry of a sperm, what happens is that there's always a rotation that happens. So the pigmented cortex is here, and then eventually there will be a gray crescent that is formed. And then future dorsal side of the egg will be this part, and then this already gives you the idea that this will be the ventral side of the developing embryo. Okay. So in chicks, gravity is involved in establishing the anterior-posterior axis. So later pH differences between the two sides of the blastoderm establish the dorsal ventral axis. Okay? And then in mammals, experiments suggest that orientation of the egg and the sperm nuclei before fusion may help establish embryonic axis. So as you see here, for the axis formation, it is not plainly what do you call this? Hindi lang dun sa position or rotation or sperm entry yung um, ginagawa nating basis of the axis formation. Like for the chicks, gravity is very important, and meron ding kinalaman yung pH. Okay? It's not just the entry of the sperm, which is very important when it comes to the axis formation. Okay? So restricting developmental potential. So Hans Penman performed experiments to determine a cell's development potential or the range of structures to which it can give rise. So embryonic fates are affected by the distribution of the determinants and the pattern of the cleavage. And then the first two blastomeres of the frog embryo are totipotent, or they can develop into all the possible cell types. Okay. So this experiment, so how does the distribution of the gray crescent affect the developmental potential of the first two daughter cells? So in here we have a control group. So when you say control group, this is the normal development. So in here, as you see, after the development of the gray crescent and then of course the cleavage, it will eventually form into two normal larvae. Okay, but in the experimental egg or the experimental group, so if, for example, if this is the gray crescent, and then there is or the cleavage of this developing embryo has been placed, kumbaga, yung gray crescent, hiniwalay siya dun sa posterior side niya. Unlike this one na yung equal right and equal left ay kinonsider. In this one, ang mangyayari, magkakaroon ng isang normal na larvae, and then the other one would just be or will just develop or will just have an underdeveloped larvae. So belly piece lang yung magiging madedevelop dun sa part na to ng zygote na hiniwalay dun sa merong may gray crescent. So it's very important that there is, the an ample amount of the gray crescent for you to be able to have a normal developing zygote or normal developing larvae. Okay? So, nagigets naman yung experiment na to, kung ano ba yung importance ng gray crescent for the developing embryo. Okay naman. So, madali lang naman intindihin yung experiment, no? Pinapakita lang dyan na yung cleavage of the different cells are also very important when it comes to its or the development of the embryo. Okay, so dapat equal yung amount nung animal pole, nung vegetal pole, and then yung gray crescent. So if hindi, of course, eventually the daughter cells would be also different from each other. One may be developed and then the other one can be underdeveloped. Okay? So, in mammals, embryonic cells remain totipotent until the 8th cell stage, much longer than the other organisms. So again, when you say totipotent, it has the possibility of developing into different types of cells. Okay? So progressive restriction of the developmental potential is a general feature of the development in all animals. And in general, tissue-specific fates of cells are fixed by the late gastrula stage. Okay? So, cell fate determination and pattern formation by inductive signals. So, as embryonic cells acquire distinct fates, they influence each other's fates by induction. Okay? So, the organizer of Spenman and Mangold, so they have, presented this, that there is a specific organizer in an axis of an animal model. Okay, so Spenman and Mangold transplanted tissues between early gastrulas and found that transplanted dorsal lip triggered a second gastrulation in the host. The dorsal lip functions as an organizer of the embryo body plan, inducing changes in surrounding tissues to form notochord, neural tube, and so on. So the inquiry is, can dorsal lip of the blastopore induce cells in another part of an amphibian embryo to change their developmental phase? Okay, so we have already discussed that there is a formation of a dorsal lip. Ito yung parang nagki-cleave, di ba? Dun sa formation ng embryo. So yung sinasabing dorsal lip na yon, siya yung kino-consider ni Spemann and ni Mangold na organizer ng mga body plan ng mga embryos. Okay? So ano nga ba yung sinasabing experiment na to? So ang ginawa lang talaga nila dito is that kumuha sila ng part ng blastopore na yon from similar species, okay? Similar species of developing embryo. Kumuha sila ng part ng blastopore na yon, and then they transplanted it to another part nung isa pang embryo. So ang nangyari ngayon, nagkaroon ulit ng panibagong development or transduction or transplantation na nangyari dun sa isang embryo na yon. So, ibig sabihin, sa loob ng embryo na yun, nagkaroon pa ng isa pang embryo. Okay? So, ganun ka-importante yung dorsal lip, okay, for the mammalian body plan or the embryo of a body plan. So, in this one, ayan, this is the experiment itself. So, this is the pigmented gastrula. of a donor embryo, and then they transplanted it to another part of the recipient embryo. So what happened is that ganto yung naging itsura niya. So pag kinross section mo siya, ayun, makikita nyo sa loob na kung ito yung primary embryo, dun sa loob niya nagkaroon pa ulit ng isa pang embryo. Okay? So nung nag-develop na sila, ganyan yung itsura niya. Ito yung donor embryo, and then ito yung recipient. Okay, ito yung donor. Okay? So, ganyan yung naging itsura na. So, primary structures, nagkaroon ng neural tube, nagkaroon ng notochord. Dun sa isa pang dinonate na embryo sa kanya, meron din siyang sariling notochord and neural tube. Okay? So, ganyan yung nangyari sa Spearman and Mangold experiment. Okay? So, formation of the vertebrae limb. So, inductive signals play a major role in pattern formation and development of spatial organization. So, the molecular cues that control pattern formation are called positional information. So, this information tells a cell where it is with respect to the body axis, and it determines how the cell and its descendants respond to future molecular signals. So the wings and the legs of chicks, like all vertebrate limbs, begin as bumps of tissue called the limb buds. Okay, so doon nag-uumpisa lahat ng mga parts or different organs of the body. Nagkakaroon ng bulge. Okay, so for the wings and the legs, you call it the limb buds. So, in this one, yan, ito yung itsura. So, from a developing embryo, magkakaroon ng mga tinatawag nga na bumps. That's what you call the limb bud. Okay? So, what are important for you to know about the different signals or yung mga tinatawag na intracellular signaling, yung mga meron tayong mga AERs, okay, mga ZPAs, okay, mga nakikita yan sa mga formation ng limbs. Okay, sila yung mga parang chemical signals, okay, na kailangan for your limb formation. Okay? So, kapag meron tong mga to, magkakaroon ka na ng apical ectodermal ridge. Okay? So, dyan na mag-uumpisa yung pagde-develop ng mga limbs. So, the embryonic cells in a limb bud respond to positional information indicating location along the three axes. Again, proximal distal, anterior posterior, and the dorsal ventral. So, one limb bud regulating region is the apical ectodermal ridge. Okay, so the AER is thickened ectoderm at the bud's tip, and the second region is the zone of polarizing activity or the ZPA. So the ZPA is a mesodermal tissue under the ectoderm where the posterior side of the bud is attached to the body. So the tissue transplantation experiments support the hypothesis that ZPA produces an inductive signal that conveys positional information indicating posterior. Okay, so ibig sabihin si ZPA, more on sa mga direction niya, more on the posterior side siya. Okay? So, in this one, as an example, we have the wing of the chick embryo. So, again, we have the anterior, posterior, ventral, and distal, proximal, and the dorsal sides of the wing. Okay? So, kung saan yung distal, yun yung mga digits, yung mga anterior, ayan. So, dapat alam nyo na to. So, another inquiry, what role does the zone of the polarizing activity play in limb pattern formation in vertebrates? So, in here, we have again an experiment. We have the donor limb bud. And then, nilagay itong ZPA, no? Nilagay na naman siya to another one. Ang nangyayari ngayon, nagkakaroon siya ng dalawang protrusions. Kasi nga, for a limb bud, it is just known that you have one, diba? Which indicates a posterior linkage para makapag-produce or ma-support niya yung apical ectodermal ridge. Okay? But since magkakaroon ka ng another site dun sa iyong limb bud na magkakaroon ng ZPA, ganito ngayon ang nangyayari. Okay? So, instead na isa lang yung napoproduce, ang nangyayari, nagkakaroon ng isa pang protrusion or nagkakaroon pa ng isang part of the same organ. Okay? Yan. So, that's the result of the zone of polarizing activity. So, for the pattern, dumodoble siya. Okay? So, next, we have the sonic hedgehog. It is an inductive signal for the limb development. And you also have the Hox genes, which also play roles during the limb pattern formation. So, sila yung mga important na mga inductive signals for the limb development. So, cilia and sulfate. So, ciliary function is essential for proper specification of sulfate in the human embryo. So, motile cilia play roles in left or right specification, and monocilia or the non-motile cilia plays a role in normal kidney development. Okay? So, next, we have here an application, which is the situs inversus. It is a reversal of the normal left-right asymmetry in the chest and the abdomen. So, more likely, this is the normal location of your internal organs. No? But if you have, situs inversus, the formation or the position of the internal organs of a human is, on the different, it's actually on the opposite side of the body. Okay, so this is how important it is for you to know, how the signaling and what are the mechanism of actions of these ZPAs, AERs, okay, and then this inductive sonic hedgehogs and the Hox genes, kung paano yung movement of cells. Okay? So that actually ends my